So we are going to the next speaker, who is Evangelos Mourkas from University of Bath, who will be talking to us about the ecology of interspecies recombination among the zoonotic bacterium Campylobacter. Very dramatic, I like it.
Hello, just make sure that all the technical parts are working. So yeah, I'm Evangelos. I'm doing my postdoc at the Sam Shepherd's group at the University of Bath. And this project I'm going to talk about is one of my PhD projects. Actually, it's the only one that hasn't reached the publication status yet. But yeah, let's just go straight into it. Right. So for horizontal gene transfer. So we know that horizontal gene transfer can allow traits that have evolved in one bacterial species. Where is my laser point? Anyway, oh wait, there we go. Right, so horizontal gene transfer can allow traits that have evolved in one bacterial species to transfer to another. So this has potential to promote new adaptive trajectories, such as zoonotic transfer or antimicrobial resistance. So the probability of horizontal gene transfer is influenced or governed by the interaction of multiple factors. So if you can call them functional phases, and it can only occur when there are gaps that align in each successive ecological, mechanistic, or adaptive barrier within a given time frame. So where am I going with this? So Campylobacter. Campylobacters are an ideal subject to study how ecology influences the maintenance of genetically distinct species. So first, they're a common component of the commensal gut of mammals, birds, and reptiles. And since they have a poor survival outside the host, that creates island populations and ecological separation. So we investigated the role of ecological barriers to interspecies gene flow in over 600 genomes of around 30 different species. So we saw that there are species and strains that inhabit one or multiple hosts, possibly occupying different subniches within the host. And then we also saw that there are over 12 species that they are identified as human pathogens. So we know from previous work from the group and others that there is an admixture, interspecies admixture between C. jejuni and C. coli. And that suggests that host ecology plays a role in the maintenance of species. So we looked at the core and the accessory genome in all these Campylobacter species. And when looking at the core, we saw an elevated average nucleotide identity between Campylobacter jejuni and Campylobacter coli, which we already knew. And that provided some initial evidence of interspecies gene flow, which we've seen before. But when we looked at the accessory genome recombination in several species, this was inconsistent with the phylogeny. And this is an example here of these species that I will never pronounce correctly. This Campylobacter laninae. So this much of the accessory genome was shared with other species. So we quantified this recombination among different Campylobacter species in isolates that originate from the same host and also from different hosts. So in each case, we wanted to see if Campylobacter jejuni that lives in a cow shares more DNA with the Campylobacter coli that lives in a cow than Campylobacter jejuni that lives in a chicken. And then we divided our isolates in different donor and recipient groups to test this hypothesis. So in the groups that the results proved the hypothesis, we saw interspecies recombination. So for example, Campylobacter jejuni lineages chloride complex 21 and 45, also Campylobacter coli ST828 in chicken. We saw Campylobacter DNA from C. jejuni CC45 was introduced in other species, so in Hepaticus, Concisus, and Ureolyticus, and we also saw jejuni DNA from cattle. So basically, in every pair, we saw elevated more recombination within the same host rather than between hosts. Right, so in the end, we wanted to come down to genes, and we wanted to focus from the previous analysis on genes that were highly recombining among different pair combinations. So if you think that bacteria inhabit the same niche, they benefit from functionality conferred by similar gene combinations. So recombination promotes the spread of adaptive genetic elements. So we postulated that genes which recombine in different Campylobacter species may be beneficial in multiple genetic backgrounds. So these are just two, they look fancy and a bit complicated, but all they show is that highly recombining genes from the previous analysis. And there is a preprint of all this if you want to go and look in a bit more detail. I haven't kept track of time. I hope I'm okay. And you've seen the same picture before from Jess. Loads of people to thank, the group, collaborators, and you for listening. I don't know if there's time for questions.
Thank you, Evangelos. That was a really interesting talk. I really enjoyed that. I think we maybe have time for one question, if that's okay. And then if anybody else has questions, we can talk later in Discord. We have one question. You show some evidence of sharing between Campy species. Do you think it is likely that there is sharing between genus?
Yes, I mean, one of the typical examples is AMR. So we've seen AMR genes across genera. So we know there's movement. So yeah, that's one example. Trying to find where Discord is in all my tabs.
Okay, I think if that's it, we'll move on. And our next speaker is Samuel Bloomfield from the Quadram Institute. Thank you.
In poultry production, we can observe a wide diversity of Campylobacter jejuni. However, for a specific broiler chicken flock, a single Campylobacter jejuni strain is often isolated from the ceca of chickens. And in the recent study, authors show the presence of a dominant Campylobacter jejuni strain in liver, but sometimes this strain was different from that found in ceca. So we have speculated that good colonization and extraintestinal spread to the liver by Campylobacter jejuni in broiler chickens are strain specific and differ when a multi-strain colonization occurs. Previously, in our laboratory, we have identified and isolated different Campylobacter jejuni strains with varying colonization abilities in the ceca after 7 days of colonization. And in this study, we used two Campylobacter jejuni strains, strongly competitive strain S and weakly competitive strain W that we characterized previously. And in this study, we had 2 experimental rooms. And in the first room, we had 4 chickens group, not infected mean 10^3 CFU of the W strain, infected mean 10^3 CFU of the S strain, and infected mean 10^3 CFU of both strain S and W. And in the other room, it was the same experimental design, but we decided to increase the inoculum of the W strain to check the effect on competition between strains. And all birds were infected at 14 days old, and we euthanized birds at 7 dpi and 21 dpi, and we collected intestinal content second and inhale to count total Campylobacter jejuni and to perform a qPCR strain specific. And we collected liver to count total Campylobacter jejuni, and because of a low amount of Campylobacter jejuni in liver, we performed PCR strain specific from isolated colonies. And because of time, I'm going to present only qPCR and PCR results. So in the first room, in cecal content, we observed at 7 dpi and 21 dpi by bacterial culture and by qPCR that the W strain didn't colonize birds, unlike S strain and Bos strain. And in birds infected by Bos strain, we observed that S strain were more abundant at 7 dpi. And in the same group at 21 dpi, we observed that an increase of the W strain close to the S strain. And in mineral content, we observed the same result. Moreover, we observed an extraintestinal spread to the liver of the W strain only at 21 dpi in birds infected by both strains. With result, it seems that W strain benefits from the S strain colonizations for its good colonizations and its extraintestinal spread to the liver. And in the other rooms, in cecal contents, we observed at 7 dpi and 21 dpi, all birds infected were colonized by Campylobacter jejuni. And in birds infected by both strains, we observed a close amount of both strains. And in the same group, at 21 dpi, we observed S strain were more abundant. And in mineral content, we observed the same result. Moreover, we observed an extraintestinal spread to the liver for the W strain only in birds infected by Bos strain at 7 dpi and 21 dpi, and in birds infected by W strain alone at 21 dpi only. With results, it seems that both strains compete in the cecal, and the presence of the S strain would accelerate the extraintestinal spread to the liver of the W strain. So in conclusion, we have confirmed strain-dependent cecal colonization and strain-dependent ileal colonization and hepatic dissemination by Campylobacter jejuni, and we have identified for the first time a commensalism relationship between two Campylobacter jejuni strains for intestinal colonization and dissemination to the liver. And these results highlight the importance to limit the introduction of new Campylobacter jejuni strains by implementing effective security measures to avoid the amplification of good colonization and liver dissemination of strains already present on the farm. And I thank all members of my laboratory, all grants fund, and you for your attention.
Thank you, Sophie. That was a really great talk. I really enjoyed listening to that. I might ask one quick question before we move on. I was wondering if you had any data regarding phenotypic differences in vitro between the two strains, the weak and the strong, that could explain the differences in the animal model?
I started to check properties in vitro by co-culture of both strains, and I saw when both strains were together, strains grows faster.
Okay, thank you. If there's any more questions for Sophie, could you direct them to Discord for afterwards? Moving on, we have a talk from Małgorzata from Warsaw University of Life Science. There is no slides for this talk, so we should move on. Małgorzata, are you here?
Good afternoon, everybody. I would like to share the results of our study on occurrence, virulence potential, antimicrobial resistance, and genetic diversity of Campylobacter strains of canine and feline origin. We examined 222 fecal samples of shelter and private origin, and the prevalence of Campylobacter in these animals were 13%. Out of 34 strains, we isolated from 29 animals. 22 of them were identified as Campylobacter jejuni, 9 were identified as Campylobacter nari, and 3 were identified as Campylobacter upsaliensis. The prevalence of genes responsible for motility, CLA and CLA-B, and cytolethal descending toxin production, CDPA, B, and C, were 100%, as well as for 3 of 4 genes responsible for adhesion and invasion. However, we did not detect KDF gene in 3 Campylobacter upsaliensis strains. 91% of the strains were resistant to ciprofloxacin, 22% were resistant to tetracycline, and 76% were resistant to ampicillin. We did not detect any resistance to erythromycin and gentamicin. In all ciprofloxacin-resistant strains, we detected point mutation resulting in amino acid substitution. In resistant strains, we detected the TET-O gene on the plasmid DNA. 85% of the strains were resistant to 2 or more classes of the antimicrobials, and almost 12% of the strains were classified as multi-drug resistant. For genetic diversity of the chosen strains, we performed post-field gel electrophoresis on 9 isolates of Campylobacter jejuni of shelter origin, and we compared them with a human reference strain of Campylobacter jejuni. Examined strains clustered into 6 pulsotypes, whereas pulsotype G02 consisted of 3 strains and pulsotype G05 consisted of 2 strains. The remaining pulsotypes consisted of single strains, and the human isolate clustered into different pulsotype than canine isolates. In pulsotype G02, 2 of 3 strains shared the same antimicrobial resistance patterns with very similar MIC values. In pulsotype G05, both strains shared the same antimicrobial resistance pattern and were classified as multi-drug resistant. Based on these preliminary study results, we can carefully conclude that pets, especially dogs, may be source of diverse antimicrobial resistance and potentially virulent Campylobacter strains for humans, particularly exposed to frequent contact with those animals, especially pets owners and shelter employees. This issue, of course, needs further investigation on larger scale, considering the bigger area of the country to obtain more meaningful results. But this would be all for now, and thank you for your attention.
Thank you, Małgorzata. So while somebody will, I think everybody's now coming up with questions, I might ask a question. So you said that the strains were quite different between the owners and the dogs. In terms of kind of diversity, like how different are they phylogenetically? Are they quite still similar, but kind of different, or are they like completely different kind of specialized clonal complexes? Maybe you have mentioned it, but if you can clarify on that, it would be really good. Małgorzata? Maybe.
Should we just move on?
Yeah, I don't think she's here, but yeah, that'd be good. Okay, so the last speaker of today is Louise Hock from Luxembourg Institute of Science and Technology.
Good afternoon, everybody. I should like to start by thanking the Campy UK committee for offering me the possibility to present my work. Luxembourg presents one of the highest incidences of Campylobacter infection in the European Union, with Campylobacter jejuni confirming more than 80% of cases. Most cases were attributed to poultry, but all warm-blooded animals are considered as asymptomatic and amplifying hosts for Campylobacter. Most of the time, humans are infected by the ingestion of contaminated food. Then, sick persons shed the bacteria into the environmental water cycle via the wastewater. Water acts as a vector of transmission from the original reservoir to the next host, which could be chicken or other animals. By surface runoff, peristore, and manure spreading, animals could also contribute to the contamination of surface water. Sewage discharge and stormwater collection could lead Campylobacter to wastewater treatment plants as well, and consequently to environmental waters. The aim of the Campylomake project is to assess the carriage of Campylobacter genotypes in wild birds to enhance our current understanding of this transmission pathway and the geographic spread of some genetic lineages of clinical Campylobacter. More than 300 wild birds were collected in Luxembourg since 2019 from 43 species, including resident, migratory, and waterfowl birds. In parallel, several surface waters in Luxembourg were also sampled to investigate their contamination by same strains than wild birds. Campylobacter isolation were based on a passive filtration method, taking advantage of the small shape and high motility of Campylobacter. Briefly, there are no enrichment steps responsible of diversity reduction. The sample is placed on a filtration membrane, which are then placed upside up on a selective solid medium. Then, DNA are extracted from colonies and submitted to whole genome sequencing. The sequence type and the core genome MLST profiles are extracted from genome sequences, and genetic profiles are compared to clinical ones identified from human patients. In our collection of Luxembourg wild bird samples, 29% are colonized by viable Campylobacter jejuni. In comparison, Campylobacter jejuni is present in 15% of surface water samples collected. The most contaminated bird species are western jackdaws, carrion crow, Eurasian blackcaps, magpies, and common blackbirds. On the other side, no Campylobacter have been found in Eurasian blue tits, grey tits, European robins, or gooses. The 124 Campylobacter jejuni strains isolated from wild birds were assigned to 49 sequence types and 55 core genome MLST types or complex types. The barcodes represent the proportion of each sequence type and complex type encountered in wild birds. 39% of isolated Campylobacter jejuni belong to only 6 sequence types. Several complex types found in wild birds are known as recurring or sporadic human genotypes in Luxembourg clinical isolates. For example, the red circle corresponds to recurring genotypes, whereas the green circle surrounds sporadic one. In conclusion, even if the prevalence of human recurring genotypes is low in wild birds, some of them are carried by birds. So the carrying by wild birds could contribute to the persistence of some Campylobacter jejuni genotypes regularly found in human infection cases. The subsequent steps will consist to trace back some reoccurring lineages of Campylobacter jejuni identified in human infection and define the role of different reservoirs in their persistence over years. This gain of knowledge will be helpful to guide and reduce the public health impact of this pathogen. Many thanks for listening, and please contact me if you have some questions.
Thank you for that talk. It was really interesting. I might ask one question. I was wondering if there is any correlation between the Campylobacter positive surface water and distance from agricultural lands?
Hi, thank you for the question. To be honest, we don't observe that, so I'm not sure for the answer.
Okay, that's great. I think if there is no further questions, we'll move on to concluding remarks from Ben and Desi.
I just want to thank all the speakers again for giving talks. Hopefully we've encountered every type of technical problem we're going to encounter today, and all the other sessions will run smoothly. And a reminder that tomorrow morning we'll start at 10 o'clock with Ozan's pathogenesis session, and then Nicole's public health epidemiology session in the afternoon. Can I remind everybody that there's questions are all on the Discord, and you can carry on chatting to all the different speakers there, and hopefully there'll be a lot of engagement. And I'll see you all again tomorrow. I'm going to hand over to Desi to tell you a little bit about her Chicken Journal Club that has been running during the term time. Over to you, Desi.
Thank you, Ben. Hi, everyone. Just wanted to plug in the Chicken Journal Club. I have put it on Discord as well, so just wanted to just talk a little bit about it. But so yeah, Chicken Journal Club has been going on now over a year. I'm one of the co-founders. We have a lot of great admin team who have been supporting this club, but basically we are a group of more like a platform where we invite speakers who are working on poultry related research. What we really wanted to do was to be able to have a chance to talk to the authors, but also to have a chance to kind of be a bit more informal and be able to ask questions. So usually what we have had is a lot of talks on Campylobacter, which has been really great. You can see on our YouTube channel, we had Ben's talk. We also had Fran, who's speaking tomorrow. We had Evangelos and Ozan as well. And a lot more topics also related to different kind of interdisciplinary fields. So not just from microbiology, but we have social scientists, archaeologists who spoke about like chicken domestication. And we had chicken welfare sessions from Marion Dawkins. And we're also trying to kind of encourage more early career researchers to think about what do you want to do next kind of thing. So if you want to move from academia to policy, what can you do? We had an incredible speaker, Dr. Anush Panjwani, who came to talk to us about her path. And the other things we've been doing is things like crash courses. So these are more for like undergraduates, master students who are kind of interested in our fields, but maybe kind of maybe don't want to necessarily come into the Chicken Journal Club. So we had these sessions also available on YouTube. And for next term, we are really excited about this new event. It's called Chicken Pitch. And basically, we are going to find ways to collaborate more together. So maybe somebody has a lot of data, but doesn't know how to analyze it. So maybe we can kind of find data science collaboration, or maybe somebody has a really great idea, but doesn't know kind of how to write grants. So you can kind of work with different institutes. And just to let everybody know, we have over like 170 members who are on our emailing list. So it's been a really, really positive kind of environment that came out of this COVID pandemic situation. So we are mainly on Twitter and also on YouTube, but also we'll be launching our Instagram, Discord, and also Facebook. And our term will be starting on 11th of October. So if anybody's interested, please check out this link tree, which is on Discord. Thank you. And we will see everybody tomorrow. Thank you.